My name is Ángel Goñi Moreno, and I lead the biocomputation laboratory at the Center for Plant Biotechnology and Genomics of the Technical University of Madrid. In this center, there is a lot of activity going on regarding plant biotechnology, including synthetic biology approaches to agriculture and forestry. And there is a big part of activity as well, and this is the one we are mostly involved in, that deals with the rational engineering of microbes. And this goes for a number of different purposes, from answering fundamental questions and to biotech applications. Specifically, at the biocomputation laboratory, we are interested in, firstly, understanding the mechanistic basis of information processing mechanisms in bacterial cells, and then to try to harness the power of these information processing abilities to engineer novel information processing devices. By information processing, I mean the ability or the capabilities to transform some inputs into outputs according to some rules that are finally encoded into genetic elements and genetic regulatory networks. That pipeline, as it is perfectly consistent to the notion of computation. So at the end, you have a genetic regulatory network that performs a biocomputation that we call a genetic circuit. So one of our aims is to establish the limits of biocomputing by exploring new ways of doing biocomputing. So, for instance, we are able to engineer genetic Boolean logic gates in bacterial cells, but what else? Or we use genetic components and DNA sequences as the primary target for implementing biocomputations. But can we use the rest of the cell? Can we use the rest of the cellular machinery to this end? For instance, can we use stochasticity? Can we harness the power of the messiness of biological systems, noise, etc.? What about evolution? Evolution is at the top of our agenda at the minute. Also because in conventional computers are very bad at handling evolution, no? But cellular computers and living systems do naturally evolve, of course, no? And evolution, as we are looking at it, is a very powerful source for information processing in living organisms. So at the end, we aim at including all these different principles in order to engineer increasingly powerful biocomputations in living cells. For some time now, we have been researching on how to reprogram, by reprogram I mean how to modify the performance of a given biological circuit without modifying the genetic components of it. So by using the background, or what we call the context, of these genetic regulatory networks. For instance, we found out that intracellular space is a key. It's very important to determine the final performance of the circuit. So depending where you place the component, the relative distance, the relative positioning of the components inside the cell, the performance is going to change. And there are many other parameters, contextual parameters, no? The plasmid vectors, depending what plasmid vectors you are using, the performance is going to be different. The host, the species that you are using to host your devices are going to impact on the performance as well, environmental conditions and so on. So there are many contextual parameters that can influence the way a genetic circuit is going to perform. And the good thing, and what we found out, is that you can rationally engineer all that contextual parameters, so that at the end you can modify the performance of a genetic circuit without modifying the genetic components and the genetic basis of it. So at the Biocomputation Lab, therefore, we are very interested in moving away from reductionism and try to embrace biological complexity for the sake of engineering, as I said before, increasingly powerful biocomputing devices in cells. Maybe one day even more powerful than conventional computers at specific tasks, and that is what we call recently the cellular supremacy. Our laboratory is very interdisciplinary, so we use mathematical modeling and computational simulations to design and to predict what is going to happen in the lab. And then we use molecular biology and microbiology in order to build and to implement the actual genetic circuit. As part of our daily workflow, we are very interested and we are very active in automation and standardization efforts, from setting up robotic pipelines for automating circuit implementation and construction, to contributing to standardization efforts like the synthetic biology open language SBOL, or the standard European vector architectures, the SEBA standard. And within this framework is that we collaborate with the European project BioRoboost. So in the biocomputation laboratory, we are always very happy to engage in new collaborations and even in discussions. So please feel free to contact us. Thanks.